When you think of the greatest engineering marvels in human history, your mind probably goes straight to things like, I don't know, the Apollo missions or maybe building the International Space Station.
Right, yeah. The really big, visible stuff.
Exactly. You picture thousands of scientists, decades of meticulous planning and, massive factories churning out these highly specialized parts.
Because it's the pinnacle of conscious coordinated effort. I mean, every single thruster, every bolt, every circuit board has a very tangible origin story
Yeah.
that you can trace back to a specific blueprint.
But today, we are going to explore an engineering marvel that makes the space station look like a child's plastic building block set.
It really does.
And the wildest part is that this massive construction project happens completely out of sight. So welcome to this custom-tailored deep dive designed specifically for you. Today, we are mapping the invisible 280-day journey from conception to birth.
Which is just a staggering timeline when you think about what actually gets accomplished.
Oh, absolutely. We're going to track how two solitary microscopic cells somehow manage to, assemble themselves into a fully functioning, sensory-aware human being.
And we're pulling this from Jenny M. Wagner's incredibly detailed textbook chapter on conception and fetal development.
Hmm.
Because this isn't just basic biology, it is genuinely the ultimate masterclass in project management, adaptation, and timing.
Okay, let's unpack this. Our mission today is to extract the absolute most mind-blowing biological engineering feats that happen along the way. So let's start at the very beginning with the raw materials.
Yeah, to really grasp the magnitude of this journey, you have to look at how those two starting materials are forged. It all comes down to a fundamental mathematical problem the body has to solve regarding cell division.
Right, because most of the cells in your body, whether they're like skin cells, liver cells, muscle cells, they replicate through a process called mitosis.
Exactly.
And mitosis is basically just cellular cloning. So a body cell has a full set of 46 chromosomes. It makes an exact copy of its DNA, and it just splits into two identical daughter cells.
Which is a perfect system if you scrape your knee and just need to patch it up with more skin.
But using that exact cloning process to create a new human would result in absolute biological chaos.
Oh, total chaos. I mean, if we simply combined a standard 46 chromosome cell from one parent with a 46 chromosome cell from the other, the resulting cell would have 92 chromosomes,
Wow.
right? And then the next generation would have 184. The genetic instructions would just become so overloaded the organism couldn't even survive.
So the body needs a completely different mechanism for the reproductive cells, the germ cells, just to prevent this massive mathematical pileup, which means it has to somehow have the genetic material before the race even starts.
Yes, and that mechanism is meiosis. So instead of just copying and dividing once like in mitosis, the germ cells replicate their DNA and then they divide twice.
Wait, twice?
Yeah, twice. They reduce their chromosomal count from the diploid number, which is 46, down to the haploid number, which is 23.
Okay.
So by the end of this process, a mature egg and a mature sperm each carry exactly 23 single chromosomes.
But wait, hold on. If the goal is just to get to 23, why go through the whole complex process of doubling and then dividing twice? Like, why not just split the original 46 down the middle from the start?
Well, what's fascinating here is that double division process is the very engine of human diversity.
How so?
Because during meiosis, the chromosomes don't just politely separate into two neat piles. They actually actively intertwine, they swap segments of DNA, and they distribute the genes entirely at random.
Oh, so it's a massive genetic reshuffling.
Exactly. This randomized mixing of alleles is what guarantees that, barring identical twins, no two siblings will ever be exactly alike. Even though they're built from the exact same two parental source codes, it ensures evolutionary variety.
It's nature's way of shuffling the deck every single time. And, once those unique cells are forged, the sheer physical scale of the journey to bring them together is just staggering.
It's an epic journey.
Just looking at the sperm's journey alone, anywhere from 100 to 500 million sperm begin this race. That is 500 million.
Yeah, and to put that in perspective, they are swimming at a rate of roughly 2 to 3 millimeters per minute.
Which to us sounds completely imperceptible.
Right, but on a microscopic scale, you have to realize they are fighting against the highly acidic downward currents of the female reproductive tract.
Uh-huh.
The distance they must cover is roughly equivalent to a human swimming across the entire Pacific Ocean.
That is a brutal gauntlet. Because out of 500 million runners, Only about 300 to 500 actually survive to reach the arena of the fallopian tubes where the egg is waiting.
And the craziest part is that the survivors can't just cross the finish line as they are.
Right.
They have to undergo this really intense, on-the-fly physiological transformation called capacitation.
Because the sperm arrive wearing a protective biochemical coating over their heads,
Right.
which was necessary to survive that highly acidic journey.
Exactly. But that exact same coating blocks them from fertilizing the egg. So capacitation is the process where the reproductive tract's fluids chemically strip that protective helmet away.
I was trying to visualize this when looking at the text, and it feels exactly like an ultramarathon where the runners are sprinting through a blizzard in heavy winter coats.
Oh, that's a great way to put it.
But to actually be allowed across the finish line, they have to literally rip off their coats while at a full sprint. Because stripping away that layer is what releases the specific enzymes they need to drill into the egg's outer shell.
Right, and those enzymes, particularly one called hyaluronidase, they act like microscopic chemical drills. They break down the tough layers surrounding the ovum.
And the finish line is entirely ruthless.
Completely. The moment a single sperm successfully breaches that inner membrane and delivers its 23 chromosomes into the egg, an electrical and chemical shockwave sweeps across the surface. This is known as the zonal reaction.
The deadbolt slams shut.
The membrane instantly and permanently hardens. It becomes absolutely impenetrable to the hundreds of other sperm that survive the journey and are, still trying to drill their way in.
So it's a strict winner-takes-all scenario.
It is. The genetic material fuses, that magic number of 46 chromosomes is restored, and conception is achieved. We now have a single-celled zygote.
The race is won, but there is zero time to celebrate because the solitary cell is just floating in a void and it has to immediately set up camp and build its own life support system from scratch, or it will die.
Yeah, the timeline is incredibly tight. Over the next 6 to 10 days, this rapidly dividing ball of cells slowly travels down the fallopian tube into the uterus.
And to survive, it has to burrow into the nutrient-rich uterine lining, which is a process called implantation.
Right. And during this window, the outer layer of the cell mass, which is known as the trophoblast, begins aggressively laying the foundational infrastructure for the placenta.
And we have to talk about the placenta because it is just an absolute powerhouse of biological engineering.
It really is.
It essentially acts as a temporary multi-tool organ. It takes on the role of the fetal lungs, the kidneys, the digestive tract, and the endocrine system all at once.
It manages a phenomenal workload. I mean, in those precarious early days, the placenta begins manufacturing a hormone called human chorionic gonadotropin, or HCG.
And that's the hormone that triggers a positive result on a pregnancy test, right?
Exactly. But it's not just a marker.
Yeah, I always thought of HCG as just like a byproduct, but it's actually an active communication system, isn't it?
It is a vital chemical text message to the mother's body. Normally, a woman's body would prepare to shed the uterine lining. The HCG hormone overrides that system.
Yeah.
It signals the body to keep producing the estrogen and progesterone necessary to maintain the lining, keeping the pregnancy viable until the placenta grows large enough to take over that hormone production itself.
It's an incredible biological failsafe. And connecting this sprawling life support hub to the developing fetus is the umbilical cord.
Right.
Now, biologically, we normally think of arteries as the vessels carrying rich, oxygenated blood to our organs and veins carrying the depleted blood away. But the umbilical cord flips this entirely backward.
It does, yeah. It operates in reverse to our postnatal anatomy.
Yeah.
In the umbilical cord, the two arteries carry the deoxygenated blood and metabolic waste products away from the fetus to be processed by the placenta.
Okay, so the arteries take the waste away.
Yes. And the single umbilical vein is the vessel driving the freshly oxygenated, nutrient-dense blood back into the fetal body.
And to make sure this vital three-vessel lifeline doesn't get kinked or tied in a knot, because eventually the fetus is going to be doing somersaults in there, the vessels are encased in this thick, specialized connective tissue called Wharton's jelly.
Which is absolutely crucial. Without Wharton's jelly, the fetus could easily cut off its own oxygen supply by, say, compressing the cord against the uterine wall or tangling it.
So the jelly acts as a firm gelatinous armor that keeps the vessels open and flowing no matter how much twisting occurs.
Exactly.
But wait, hold on. I need to push back on this whole placental exchange process. We're talking about the placenta providing oxygen, transferring nutrients, and even passing along the mother's immune antibodies. But the text notes it is a strict biological rule that the maternal blood and the fetal blood never directly mix. If the blood supplies never physically touch, How are massive molecules like immune antibodies crossing the border?
It seems like a paradox, doesn't it? But it is solved by the brilliance of the placental membrane.
Okay, how does that work?
Picture the maternal blood pooling in these microscopic caverns called intervillous spaces.
Okay.
The fetal blood vessels grow down into these pools like the roots of a tree dipping into water. They are entirely surrounded by the mother's blood, but they are sealed inside a membrane that by the 5th month of pregnancy thins out to be only a single cell layer thick.
Wait, really? So there is just one microscopic cell wall separating the two entire circulatory system?
Exactly one layer. So for small things like water or basic electrolytes, they just passively diffuse right through that ultra-thin wall.
Makes sense.
For essential nutrients like glucose, the placenta uses active transport, physically pumping them across. But to move those massive maternal immunoglobulins, the antibodies that will give the newborn its initial passive immunity, the membrane utilizes a mechanism called pinocytosis.
Pinocytosis. I've heard that translated as cellular drinking, but that sounds like the cell membrane is literally acting like a mouth.
Well, actually, that is a very accurate way to visualize it. The membrane physically reaches out. It engulfs the giant antibody molecule from the mother's blood and pinches off to form a little protective bubble or vesicle around it.
Wow.
Then it drags it across the interior of the cell and spits it out intact into the fetal bloodstream.
That is genuinely incredible. It's a highly active, deliberate smuggling operation that passes along vital immune defenses without ever allowing a single drop of maternal and fetal blood to mix.
The engineering of that border control is absolutely flawless.
Okay, so we've got the life support system running at full capacity. Now, this amorphous cluster of dividing cells has to start assigning specific jobs to specific cells to actually build the physical body. It is time to roll out the blueprint.
And this architectural phase kicks off astonishingly early. By the 3rd week after conception, the cells begin to fold and differentiate into 3 distinct primary germ layers.
The ectoderm, the mesoderm, and the endoderm.
Right. And every single tissue, bone, and organ in the human body originates from these 3 foundational layers.
It's like sorting the raw materials at a construction site before you start building. The ectoderm is the outer layer that eventually becomes the skin, the hair, the nails, and the entire nervous system.
That's correct.
The mesoderm is the middle layer that will form the heavy structural components like bones, muscles, and the cardiovascular system. And the endoderm, the inner layer, folds inward to form the lining of the respiratory and digestive tracts.
And out of all these developing systems, the cardiovascular system is the first to actually power on and begin functioning. By the end of the 3rd week, before the mother even misses a period, a primitive microscopic heart is already beating.
See, here's where it gets really interesting to me. The cardiovascular system is up and running, pumping blood around this tiny body. But the fetus isn't breathing air yet, and it isn't eating food.
No, not yet.
So its lungs and its liver are essentially deflated, dormant organs under heavy construction. Pumping a massive volume of blood into them would be pointless.
Or it would be incredibly inefficient and potentially damaging. The placenta is already handling the job of oxygenating the blood and filtering waste, which is what the lungs and liver will eventually do.
So the body engineers these brilliant circulatory bypasses, or shunts. There's the ductus arteriosus, the foramen ovale, and the ductus venosus. I was looking at how these work, and it is perfectly analogous to a city's temporary traffic detour.
Also,
Well, imagine a city is building 2 massive new superhighways, the lungs and the liver. While they're actively under construction, you absolutely do not want heavy commuter traffic driving through those messy, unfinished zones.
right, that would be a disaster.
So the biological engineers build these 3 temporary off-ramps that efficiently route almost all the oxygenated blood entirely around the lungs and liver, delivering it straight to the brain and the upper extremities where it's desperately needed for rapid development.
The traffic detour analogy is spot on. These shunts ensure the brain gets priority delivery of the best oxygenated blood. But to make this whole system work in a womb, the fetus utilizes another brilliant chemical hack.
Oh, fetal hemoglobin.
Yes, fetal hemoglobin.
How does that actually differ from the hemoglobin circulating in our adult bodies right now?
Well, a fetus exists in an environment with significantly lower oxygen tension compared to the outside world. If it used adult hemoglobin, it would suffocate. So to compensate, fetal hemoglobin is chemically structured to act like a sort of oxygen magnet. It has a supercharged affinity, allowing it to carry 20% to 30% more oxygen than the mother's hemoglobin.
That's a huge difference.
It is. When you combine that highly efficient hemoglobin with a blistering fetal heart rate of like 110 to 160 beats per minute, the fetus becomes a master at extracting and circulating every possible molecule of oxygen the placenta provides.
The efficiency is staggering. Okay, so fast forward to week 9. The foundational blueprint is fully laid out. All the major organ systems are present, and the embryo officially graduates to being classified as a fetus.
Yes, the fetal stage.
From here on out, the rest of the pregnancy seems like an extended, highly choreographed dress rehearsal for life on the outside.
That's exactly what it is. The structures are built. The rest of the time is just spent practicing how to use them.
Hmm.
For instance, the fetus begins actively swallowing amniotic fluid.
Now wait, by the second trimester, that amniotic fluid is primarily composed of fetal urine?
It is, yeah.
Which, I mean, sounds completely bizarre and slightly gross to us, but it's totally sterile and serves a massive developmental purpose, right?
Swallowing that fluid is absolutely critical. It forces the gastrointestinal tract to practice peristalsis,
Mhm.
the rhythmic muscle contractions required for digestion.
Ah, I see.
It's through processing this fluid that the fetus eventually builds up meconium, which is that dark, cateary substance that will become its first bowel movement after birth.
And the sensory development during this dress rehearsal is wild. So, what does this all mean for you, the listener? By the 5th month, the taste receptors are fully active. Flavors from the mother's diet actually cross over and flavor the amniotic fluid.
They do.
And the fetus actually reacts to it. Like if the fluid tastes sweet, they swallow faster. If it tastes bitter, they slow down. They can even hear muffled sounds through the fluid and they habituate, meaning they stop startling to repeated outside noises.
If we connect this to the bigger picture, you realize they're also constantly practicing the mechanics of breathing. They make rhythmic respiratory movements, pulling amniotic fluid into the lungs and pushing it back out.
Wait, they breathe the fluid.
Yes, and this fluid movement actually helps develop the chest wall muscles and regulates the volume of the developing lungs.
When you realize that a baby is tasting the mother's food and learning to ignore the sound of the family dog barking while still in the womb, it completely changes how you view those 9 months.
Oh, entirely. The fetus isn't just passively floating in some dark deprivation tank. It's actively training its nervous system based on cues from the outside world.
It's all rehearsal.
It's entirely about survival. This in utero rehearsal ensures that the complex neurological pathways required for swallowing, digesting, and breathing are firmly hardwired before the baby ever faces the shock of taking its first breath of air.
And preparing for that first breath involves a critical piece of chemical engineering in the lungs regarding surface tension, doesn't it?
Yes, a massive hurdle.
Right, because if you've ever tried to pry apart two wet pieces of plastic, you know they stick together fiercely. Inside the lungs, there are millions of tiny air sacs called alveoli, and they are wet.
Exactly.
Without some kind of intervention, the surface tension would make them stick flat shut, and a newborn wouldn't have the chest strength to rip them open to breathe.
To solve the wet plastic bag problem, the developing lung cells secrete substances called surfactants. Think of surfactants like dish soap.
Dish soap!
Yeah, if you put a drop of dish soap in water, it breaks the surface tension immediately. Surfactants coat the inside of the alveoli, preventing them from collapsing and sticking together when the baby exhales.
And medical professionals actually track this development using something called the LS ratio, right?
They do. The LS ratio stands for lecithin to sphingomyelin, which are two specific types of surfactants. As the lungs mature, the amount of lecithin spikes significantly.
Okay, so they're looking for a specific number.
Right. When that LS ratio hits 2.1, which typically happens in the middle of the third trimester, it is the biological green light.
So it means the fetal lungs have produced enough dish soap to successfully expand and function outside the womb.
Exactly. Without that chemical preparation, surviving that first breath would be physically impossible.
It is just layer upon layer upon layer of perfect timing, which of course brings us to the vulnerabilities. Because a 280-day construction project this complex, with millions of moving parts, is incredibly delicate.
It is highly sensitive.
Any slight disruption in those early stages can completely alter the blueprint, sometimes resulting in multiples and sometimes exposing severe vulnerabilities.
Yeah, looking at multiples first, there are two primary pathways. Dizygotic, or fraternal twins, occur when two entirely separate mature eggs are released and fertilized by two completely separate sperm.
So it is quite literally two independent construction projects happening side by side in the same location.
Exactly. They share a womb, but genetically they're just standard siblings who happen to be the exact same age.
Okay, and the other type?
Monozygotic twins, on the other hand, start as one single fertilized ovum, one zygote, that spontaneously splits into two distinct embryos. Because they originate from the same fertilized egg, they share the same sex and the same core genetic blueprint.
Which is why we casually call them identical twins. Now, I have always assumed that identical twins were literally carbon copies of each other, like 100% identical down to the very last strand of DNA. But digging into the biology of the source text notes, that's actually a massive myth.
It is a huge misconception. When that single zygote splits, the replication process is rarely mathematically flawless.
Oh, really?
Yeah, as the cells divide to create the second embryo, tiny genetic mutations or copying errors frequently occur. So one twin could end up with a slight genetic variant, perhaps a predisposition to a certain condition, or a minor physical anomaly that the other twin completely lacks.
So they're not perfect clones?
No, they're profoundly similar, but calling them perfectly identical ignores the reality of how messy cellular division can actually be.
That completely blew my mind. But the biology also reveals what happens when the outside environment disrupts this delicate division process. And here we're talking about teratogens.
Right. Teratogens are environmental threats that cross the placenta and interfere with fetal development. This category includes certain viruses, alcohol, specific medications, and environmental chemicals.
And the golden rule of teratogens is that timing is everything, isn't it?
Timing is everything.
Because the damage they do depends entirely on what part of the blueprint is being built on the exact day of exposure, and the first 3 months, the first trimester, is the most dangerous window.
During that embryonic period, the cells are undergoing their most rapid multiplication and differentiation. They are laying the physical foundations for the heart, the limbs, the organs.
Right.
If a teratogen enters the system, while a foundational structure is just beginning to form, the interference can cause catastrophic, irreversible physical defects. Once those organs are fully formed in the later trimesters, they are much less susceptible to structural damage.
But there is one major exception. There is one organ that remains uniquely, profoundly vulnerable for the entire 280 days.
Yes.
And that is the brain.
The human brain has the longest developmental window of any organ in the body. I mean, while the heart or the kidneys might finish their structural formation relatively early, the central nervous system never stops.
It just keeps building.
It is constantly growing, constantly layering new neurons, and constantly wiring complex connections throughout the entire duration of the pregnancy.
Wow.
Because it is in a perpetual state of delicate construction, it is perpetually vulnerable to teratogenic insult. This is why cognitive or neurological impacts are the most frequent and pervasive outcomes of environmental exposure.
It really puts into perspective how mind-bogglingly precise this whole operation has to be to succeed. To wrap up this deep dive, just take a step back and look at the scope of what we've covered today.
It's a lot to take in.
We started with the math of meiosis, forging 23 solitary chromosomes that survived a race against 500 million competitors. We saw the rapid construction of the placenta, a temporary superorgan managing everything from life support to cellular smuggling operations.
The pinocytosis.
Exactly. We watched as temporary biological highways were built to route blood around sleeping organs, and we tracked the sensory awakening of a fetus actively practicing for a world it hasn't even seen yet. It truly is the ultimate 280-day engineering marvel.
The complexity of the journey is just staggering.
Thank you so much for joining us on this exploration.
And, I want to leave you with one final thought, something hidden in the biology that brings us full circle to the idea of an engineering project that continues to defy modern science.
Oh, what's that?
Think about the immune system. We know the maternal immune system is incredibly aggressive at hunting down and destroying foreign bodies.
Right, it attacks anything that doesn't belong.
Yet the fetus and the placenta act exactly like a living tissue transplant, containing genetics that are 50% completely foreign to the mother. Under any other medical circumstance, her body would immediately attack and reject it.
That's true.
Yet the placenta somehow magically suppresses or evades that maternal immune response for 9 solid months. So I'd like you to ponder this. If medical science can one day fully decode the exact chemical cloaking mechanism the placenta uses to avoid rejection, could the invisible secrets of a developing fetus ultimately hold the key to revolutionizing life-saving organ transplants for adults?
Wow. An invisible, ancient biological process that might just solve one of the most complex hurdles in modern medicine. That definitely gives you something to think about. Until next time.